Hi class and welcome to lab 6. For this lab, you guys will just be responsible for the information in the PowerPoint. You can read through the procedures and how they would be done with a partner, but for this lab you will not need to submit a lab report at all because a lot of the lab report questions, especially for this lab, are directly taken from procedures that you would be doing with a lab partner, but you are still responsible for the material that is included in this PowerPoint. A lot of it is a review from the senses chapter of the nervous system. So there will be a lot of anatomy, specifically in the eye and the ear for this one. So we'll first talk about vision. Vision involves detecting and interpreting visual stimuli by converting light energy to nerve impulses and then transmitting them to the brain. And the structures are the eye, and we're going to go over the anatomy of the eye here. The sclera makes up the white part of the eye. The choroid layer is this second layer, the red layer. That's the vasculature layer. And then the retina makes up the third yellowish layer. The retina is the nervous layer where the photoreceptors will be located. So light enters the eye through the cornea, and then it travels through the anterior chamber through the pupil, which is the dot in the eye. The iris is a series of muscles that controls the pupil's shape, so the pupil can become dilated or constricted. And then light will travel through the lens, and the shape of the lens is controlled by the ciliary body and suspensory ligaments. So the lens will focus the image on the back of the eye in the retina. Specifically, the greatest acuity, visual acuity, is at the fovea centralis because that's where you have the most cones located in your eye. And the fovea centralis is a little point in the back of the eye about the size of a pin, and it's located in an area called the macula. The optic nerve is the cranial nerve that connects to the back of the eye, and the optic disc is the area where the optic nerve connects. It's the area where you have no photoreceptors, so we call that your eye's blind spot. And it's the area where blood vessels will enter and exit the eye. So here are those eye structures and their functions that we just talked about, so you can review those. In order to regulate the amount of light, the iris, which is made up of smooth muscle, causes the pupil to constrict or dilate to control the amount of light entering the eye. And the focusing of light involves the bending of light, and that bending is called refraction. And that occurs by the cornea and the lens. Accommodation is adjustment of the lens curvature to enable focusing on near and far objects, and it is made possible by ciliary muscles, and the ability to accommodate deteriorates with age. So here's a look at the iris. In dim light, the iris will dilate the pupil, so that's a sympathetic response. So your fight or flight response will dilate your pupils. In normal light, you can see the different layers of iris muscles. And in bright light, the parasympathetic axon will cause a constriction of your pupil. And that is a parasympathetic nervous system response. For near objects, so this is a look at how the lens changes shape to focus the image on the back of the retina. For near objects, looking at a close-up object, contraction of your ciliary muscles will reduce the tension on the fibers and allow the lens to bulge or become thicker. And for focus on distant objects, relaxation of the ciliary muscles increases tension on the fibers, which will pull on the lens and flatten it. Normal shape allows focusing on the retina. Myopia is nearsightedness. That's what most of us have who can't see things far away. This is where the eye is longer than normal. The distant objects are focused in front of the retina, and it may be corrected with concave lenses. Hyperopia is farsightedness, where it's harder, more difficult to see things close up. The eye is shorter than normal. And astigmatism are irregularities that exist in the cornea or the lens. So here's a look at a normal eye and how the image is focused right exactly on the focal point in the back of the eye. Nearsightedness, myopia, nearsighted persons can see near objects clearly, but distant objects are out of focus because the focal point is in front of the retina. And then this shows the correction and how the concave lens will correct the image to be focused on the back of the retina. Farsightedness, hyperopia. Farsighted persons can see distant objects clearly, but near objects are out of focus. And this is the convex lens that again can reposition the image on the focal plane. And astigmatism is due to an abnormal curvature of either the cornea or lens. It can also be corrected for by a contact lens usually. And again, all of these corrections are trying to get the image focused on the back of the retina. The retina allows us to see in color. The layers of the retina, the outermost pigmented cells absorb excess light. Photoreceptors are your rods and cones. Bipolar cells synapse with rods and cones. And the innermost layer are your ganglion cells, which are connected to axons that will become the optic nerve. So this is a look at those different layers. Light travels in to the eye and goes to the back of the retina first, specifically this pigmented layer, and then it kind of goes back up through the rods and cones, the ganglion cells, and then in the axons to the optic nerve. Photoreceptors are your rods and cones. They respond to light, so their photopigment protein will change shape. There's many more rods than there are cones in your eye. They're connected to about 1 million ganglion cells. And there's a significant amount of convergence and summation between rods and cones and your ganglion cells. So a rod and a cone under a scanning electron microscope, they do look like more rod-shaped and also more cone-shaped. They're rod-shaped kind of more towards the top in blue, and the cone-shaped is in green here. Rhodopsin is a photopigment within rods that's more sensitive to light than the photopigment in cones. So in dim light, your vision primarily depends on rods. Rods do not enable color vision. Rods and cones are not evenly distributed on the retina. The regions of the retina farthest from the fovea centralis will have the highest ratio of rods to cones, and that's because in the fovea, you'll have the highest percentage of cones. There's 3 types of cones, red, green, or blue, that enable color vision. The brain will interpret the ratio of impulses coming from ganglion cells connected to the 3 different kinds of cones. The cones will require stronger light to be activated. And in dim light, we primarily see rods and we don't see color. Cones are what is responsible for visual acuity. And visual receptors can adapt, and this will depend on adjustment of the pupil size and adaptation by the rods. Adaptation takes longer when going from bright light to dim light. Bright light uses up the rhodopsin temporarily, and it takes several minutes to resynthesize rhodopsin. So this is why when you walk into a dimly lit room, it takes about 20 minutes for your eyes to adjust and to be able to see in that dimly lit room. Here is an animation video of the human eye, so you can download this PowerPoint and watch this on your own. The retinal detachment, the retina separates from the choroid. So these are disorders of the eyes. Symptoms of retinal detachment could be flashes of light, blurred vision, loss of your peripheral vision, and this can be surgically corrected if detected early. Cataracts are where your lens becomes opaque or blurred. The treatment of this is to actually replace the lens with an artificial one. And glaucoma is when pressure inside the eye builds up due to improper draining of the aqueous humor, and it can be treated with drugs or surgery, but if untreated, could lead to blindness. So this is a look at retinal detachment. You can kind of see the double layer peeled up here. Here's cataracts. The cataract in this person's right eye was caused by an inflammatory disease process. AMD is age-related macular degeneration, is when the retina detaches and the photoreceptors in the macula will degenerate. This is the leading cause of blindness in people over 60. And color blindness is when you just have a lack of one of those 3 types of cones. So people might not be completely colorblind. They'll just have kind of an inability to distinguish between the full range of colors, or their blues and greens might look more purple and brown. Red-green color blindness is often inherited. and it's on the X-linked, so we call it sex-linked, and it's usually inherited, or it affects males more than females. Here's what you would look like, with a picture with normal vision, with glaucoma, which is the pressure buildup, and age-related macular degeneration. Here's a test for color blindness. Hopefully you guys can all see the number 12 in this picture, but there's different tests you can do. If you Google colorblind tests on the internet, it'll show you similar pictures like this to see if you can distinguish the colors of the number separated from the other color. Someone who is colorblind would just see all of these circles would just be one color, so they wouldn't be able to see the number 12 pop out. The human eye, so here is some anatomy that you can review. The pupil is surrounded by the iris, and the sclera is the white part. Cornea is the clear outermost covering, the place you would set a concave contact lens. You cannot see the cornea, but it covers the iris and pupil. And the retina is in the back of the eye. So here's another look at some anatomy of the eye. Feel free to pause my video to review the anatomy. Test questions for a layer lab exam might include a lot of anatomy for the eye and the ear. Again, we have a blind spot because where the optic nerve attaches to the back of the eye, you have no photoreceptors in this area. So if an image gets focused on the blind spot, you would be unable to see it. This is a test of the blind spot, and you can kind of do it if you pause the video after we read through the directions. If you close your left eye and focus on the X with your right eye and very slowly move the image closer to your head and out again if necessary, somewhere between 5 and 20 inches away, the O in your peripheral vision will seem to disappear. And that's because the image of that O being reflected on that eye in that particular distance from the screen, it will be projected onto the optic disc, which is the blind spot of the eye where you have no photoreceptors. So try that out, it's pretty cool. And this is a look at the retina, and this is where the blind spot is. This is the optic disc, where the optic nerve attaches to the back of the eye. Accommodation, tugging by the ciliary muscle changes the shape. This is how your eye focuses. So to see near, the lens will become more round, and to see far, the lens will become more flat. The visual acuity, so this is a Snellen eye test chart. And if we were in lab, you would measure your visual acuity. So this is a chart you would see in an eye doctor's office. You would stand 20 feet from the sign, and 20 feet will always be the top number of the fraction that will tell you your visual acuity. And then you would read the smallest line and record the bottom number of the fraction. And the denominator, the number on the bottom, signifies what a person with normal vision could read that many feet away. So let's say you have 20/20 vision. That's normal vision. So what you can read at 20 feet is what a normal person reads at 20 feet. So 20/20 is considered normal vision. If you have 20/25 vision, so if you can only read up to line 7, that means what you read at 20 feet is what someone with normal vision could read 25 feet away. So they could be further away from the sign and be able to read line 7. So they would have better vision than you. If you can only read to line 4, 20 feet away from the sign, that means what you're reading at line 4, 20 feet away, is what someone with normal vision could read 50 feet away. So if you can only read to line 4, that's not very good because someone with normal vision could read line 4 up to 50 feet away. So that's the fraction and how it explains visual acuity. This is the fovea centralis, looking at how the rods and cones in the fovea. So the fovea centralis is where visual acuity occurs, and it'll have the highest concentration of cones. And cones are important for visual acuity because you can see here how each cone is directly attached to one bipolar and ganglion cell. And this means there's no convergence. There's a 1:1:1 ratio, meaning what each cone picks up sense-wise from any sort of image that's being projected to it it will send that image directly up to the brain. The difference is the rods, where you have 2 and maybe 3 rods converging on one bipolar cell, and then bipolar cells will converge on one ganglion cell. So what the rods are picking up vision-wise are being converged to only travel in one axon, versus the no convergence. Each cone has its own kind of direct pathway to the brain because there's no convergence. And that's why cones are responsible for seeing clearly. And you'll find more cones in the fovea centralis. Astigmatism, this is a chart that measures if you have astigmatism. If you focus right in the center, someone with astigmatism would see this result where the 12, the 1, the 2, 6, 7, 8, 9, those lines appear bolder or darker. So this chart is already showing what someone with astigmatism would see. We have a chart like this in lab where all of the lines are the same weight, so they all look the same width. And if you would have astigmatism, some of the lines would look more bolded than others. This is a really fun test for adaptation. So what I want you to do is stare at that red circle for several seconds and pause the video, at least for 30 seconds, and tell me what happens eventually to the green circle around it. So hopefully you found out, and you should really pause the video and do this, but this shows how sensory adaptation works with the photoreceptors. Eventually, if there's constant stimuli going to the photoreceptors of the eye, they'll turn off or put their attention somewhere else. So eventually, if you focus on the red dot, the green circle around the dot will disappear. Then we get to hearing. And again, for this lab, you're responsible for hearing, especially the anatomy of the ear, properties of sound and sound waves. Loudness is related to the amplitude, measured in decibels. Pitch or tone is related to the frequency, measured in the number of wave cycles per second. So this is a look at frequency and the amplitude and how they relate with frequency. Here's a look at common noises and your hearing and how it would affect your hearing. You don't want to be near a jet engine, a shotgun, and even live rock concert or wearing earbuds a lot has definite risk of permanent hearing loss. Some of the anatomy of the ear I'm going to go through right now. We went over a lot of how sound enters the ear. The pinna is the the auricle of the ear directs sound waves to the auditory canal, and that will direct the sound waves to the tympanic membrane or the eardrum. That will turn those sound waves into vibrations to the malleus bone or the hammer. Then you have an air-filled middle inner ear that amplifies the sound as it goes through 3 tiny ossicles or bones, the malleus, incus, and stapes. And these bones will be able to several-fold amplify that sound. The stapes will pass the vibrations along to the oval window from which they will enter the inner ear where the cochlea is. The auditory tube is also known as the eustachian tube that will connect to throat to help equalize pressure. And you should have a good understanding of labeling everything in this picture. The cochlea then converts the sound to action potentials. The structure of the cochlea looks like a coiled snail. When uncoiled, it's a tapered tube filled with 2 outer canals and an inner fluid-filled duct. It contains hair cells, mechanoreceptors with hair-like extensions embedded in the tectorial membrane. And the organ of Corti is where these mechanoreceptors and hair cells are in the tectorial membrane. So vibrations of the oval window will pass through the cochlear fluid as the pressure waves. Pressure waves result in physical bending of the hair cells. Different pitch sounds result in stimulation of hair cells in different regions. So we talked about this in lecture. High pitches are near the base of the cochlea, low pitches are closer to the cochlear tip. And action potentials will then be carried to the brain by the auditory nerve to the vestibular cochlear nerve. So this is a look if we were to unwind the cochlea, which is this. There's a vestibular canal, a tympanic canal on the top and bottom, and then a cochlear duct in between. And if we zoom in on the organ of Corti, this is where we see these hair cells embedded into the tectorial membrane. And these vibrations, which turn into sound waves through the fluid, will bend these hair cells. And when the hair cells bend, that bending is picked up by the nerve. And that's how we're able to hear. So this shows how the hair cells will bend based on action potentials or stimuli. The vestibular apparatus, 3 semicircular canals. These also contain hair cells that bend in response to movement. The semicircular canals sense rotational movement. The movement of the otoliths, crystals, will bend hair cells in the vestibule, which is your utricle and saccule. And this will detect the head position and linear acceleration. So you can kind of see there's also hair cells located in this vestibular apparatus, detecting linear acceleration and head bending. This is showing the head being tilted and how those hair cells will move. These are the structures and functions, again, of the ear. Deafness is loss of hearing. Nerve deafness is damage to the hair cells, and conduction deafness is damage to the tympanic membrane or bones of the middle ear. Otitis media is inflammation of the middle ear, and Meissonier's syndrome is the inner ear condition that impairs hearing and balance. This is a great animation to review the anatomy of the ear to look at. And then the last couple slides, I'll let you guys look on your own. I include them just to review some of the anatomy of the ear itself, and a lot of them are the same picture, but sometimes it's nice to see different pictures of the same structure. So I will let you guys do that on your own. And this is a model, again, that shows the cochlea and the different chambers and canals in the organ of Corti. Thanks for listening. And again, for this lab, there's no lab report due, but you are responsible for this material for the lab exam.